Yeah, hello, welcome all. We'll get started in about 7 minutes. Thank you for joining. Yeah. Yeah, hello, welcome all. We'll get started. So, Giri, we are getting started in about 3 minutes, please. Yeah.
Sure.
Yeah, thank you. Yeah. Namaskar. Welcome to this Sci'versation with me. Today we are quite honored and I'm personally elated to have Girinath Pillai, who is the Chief Scientific Officer at Zast Innovations. Before I introduce him by and large, I request all the participants to mute your microphone, please. And if you have any questions, you may, in fact, pose in the chat box over here. So, first of all, I would like to thank Giri for taking his precious time. He has been running helter skelter, with several tasks over. He's also the chairman for this drug hackathon series that Ministry of Human Resource Development has started a couple of months ago. And he has been instrumental in organizing several other events as well, especially during COVID times. He was the motive force behind organizing several series of online events, which we all participated, which many of you would understand. So is a research scientist at Nairo Research, and is a CSO and director at Zast Innovations, Bangalore. He was a Marie Curie Fellow. So soon after he finished his MSc, he had several sabbatical visits at different institutes. Then he obtained his PhD from University of Florida with crisscross collaboration with Professor Allen and Professor Carlson at University of Tartu in Estonia. They both are well-known principal investigators, well-known in Europe in computational chemistry. And his work largely dealt on mosquito repellents with USDA, United States Department of Agriculture, including the SCR and the machine learning methodologies with targeted descriptors, which has received wide attention and which has received global collaborative efforts that Giri has been associated with. So he's basically a computational medicinal chemist, and he is largely involved in various structure-based ligand drug discovery approaches. He's also involved in drug repurposing, and he has produced a good number of mathematical and statistical approaches in lieu of these particular QSAR models. And to his credit, he was also responsible researcher and collaborator and mentor for a wide number of companies, including Johnson & Johnson and Eastman in the US. He has a wonderful patent to his credit and more than 40 papers which he has published by and large. And he has about 2 PhD fellows currently working with him. He's mentoring them amid his busy schedule. And apart from the scientific positions, he's also a mentor at IIIT Kottayam and as well as Niti Aayog government schools. And besides that, he also does a lot of humanitarian efforts through Asha Foundation that he's associated over the last 10 years. So with this certain introduction, I would like to welcome Giri. Thank you so much, Giri, for taking your time. Hear from you through your wonderful research journey. If you can go through your slides and please, enlighten us. And then, we'll have a Q&A session with you. Thank you.
Thank you so much. Okay. Thank you so much. I hope I'm audible. I'm getting some feedback. Sorry.
Yes.
Oh, okay.
Yes, Giri. Yeah, please. Yeah.
Okay. So thank you so much for this giving me an opportunity. But let me take a disclaimer in the first because this is something new to me. Usually, we always try to present something scientific and start with a slide. Of course, I have some pictures as Prash already requested me, but this is going to be a challenge for me. I'll try to keep you motivated and not so boring. In some instances, it might be selling myself, but sorry, unfortunately, this is kind of a conversation where talking about experiences, what has been passed, what mistakes have been done, how it's being recovered, all those things. And I'm happy to share what is being I have went through, what from my experience rather than what I have seen. Right. So talking about as Prash rightly said, I know Prash since many years, and we were being abroad outside of India without knowing that he's in the next building for 2 weeks. So that's how it is. World is so small. We are all getting connected. The only thing we try to find out how we have to reach the right medium to get connected. And I could be get
Sure.
connected through all the possible social mediums like LinkedIn, YouTube, Twitter, and Instagram. Uh, to be honest, during this COVID time, I was not so active in YouTube and all. It was just used for my personal purpose. But later during this COVID, I thought, okay, time is a bit boring, no business, nothing much happening. So let's see what can be done productively. So I started learning. I started learning how to do YouTube videos and share things, and slowly kicked up with few meetings and all. And later I slowly came down because I have to go back to work, right? There are certain priorities we have to set as the time changes, and time management is something very key and very vital when you're trying to be very productive. Sometimes of course we cannot be productive, we might delay. So I'll go through some interesting facts with a story of few photos, and we'll go along. If it is boring, let me know. I'll change the track. So let me tell you that it's all my personal opinion, what I'm trying to express here. If there are some questions comes in later. And the slides contains pictures or videos or contents of my friends, colleagues, and photos. I have intimated them the usage, and please do not cite without their consent. And I would like to thank everybody here, whom I have listed here. If I have missed someone, apologies here, because they are who made me at least what I am now. I'm still not anywhere. I am still trying to learn, struggling, and also moving forward with the life in career, as well as in having a good work-life balance. It's nothing that we have reached somewhere. No, we are still struggling and moving on. And again, a challenging period has also come, where we are all going through. So I'm expecting everybody is staying safe and healthy. I'm also safe and healthy. So here in this particular point, I wanted to thank all the people who said no to me, because that no actually motivated me to do something. Because why they said no to me, that started ticking my head. Why they need to do that? Is there something missing in me? I have to find what is missing in me. And second was when they told lies about me. For example, today Prash very nicely introduced me, even though it was a bit lengthy. Maybe my seventh or eighth talk in one of the conference, the organizer told that Giri is having multiple publications in peer-reviewed journals. Of course, I was not having anything. I didn't have any PhD that time. I felt bad. Why? Even I didn't write and give a write-up to them. So I was surprised why it was being done. So it was something that is being expected by the audience, or to cope up with the other speakers. So these things actually have motivated me. Something is missing in me, so I have to improve that. So take criticisms, and the people who are going to being very critic, they are the real superstars in your life. If you are going to get encouraged on trying to overcome that missing. But if you get more depressed and discouraged of that criticism, then you will be down. You shouldn't be. Of course, I was also down, very much, maybe for 24 to 48 hours. But later, it made thinking, why should we be down? What we did, something we were not overcome, or we didn't know about it. So we have to get through it. How can we do that? That's where the search starts. Until you ask something, nobody's going to give you. Nobody's going to search where is Giri and going to give me something. No, we have to go behind it. That's where now the era, the particular era or IoT era or AI era or Industry 4.0 era is something that we are selling ourselves in every medium, whatever possible. No wrong in doing it if it is not annoying people, if it is not spamming people. because how people will get to know about you, right? So that's where I am really, really thankful to them, and they're always in my prayers, because just I am here only because of them. And second comes the support of your near and dear ones, Nit and Ken. Of course, my spouse, my partner, and my kids, my parents. Of course, they took a lot of effort to have a life balance, because my PhD, major chunk of my PhD happened after my marriage, just after my marriage. So it will be challenging, right? My marriage proposals things all happened when I don't have a job, right? I was a project fellow, and I don't know, somehow they had trust on me, especially my in-law, my wife's father. So he had so much trust on me. He was just asking how much time you're going to take to complete your PhD. I said, maximum 5 years. Okay, 5 years, we are strong enough, we are live enough. That's enough for me. But are you sure after 5 years you're going to get a job? I said, yes. So that was a trust that he had on me. So that was so supportive and encouraging. And uh, to be during the day of my marriage, that's where I'm getting the offer from University of Florida. And about 48th day from my marriage day, I've flown to US, to University of Florida. But of course, before that, interviews, all those things were being happening. But somehow it was, my wife seems to be so lucky to me then. But later, the work-life balance is going to be affected, because, of course, I have to go home, spend time with her, and then that gets affected. So we all went through them with their support. If they were not supporting, I couldn't have completed my doctoral studies or research, or whatever Prash just mentioned about peer-reviewed publications, patents, and for industrial projects. Nothing would have done, because I need more time to be on work than to my social or being with my family. But still, we were able to do that during weekends. But during weekdays, it was a bit challenging. And also, teachers, especially from my school, I was not so great in school. They used to, they used to, my teachers used to shout at me, "You're not studying, you're not good for anything, you simply ask questions." Uh, it all happened, right? And when I came for my plus two, also, yeah, somehow science went in. That's how it moved on. So teachers are also, from my schooling, it is being really encouraging. And when it comes to my parents, actually, uh, especially my mom, she was so, what to say, uh, so adamant that I have to go to an English medium, not a government school. I don't say government schools are bad, they're really good. I know some of them where I mentor, they're really good, and but somehow, thoughts of parents, we cannot blame them. So my dad was, he's not getting great marks, so why should we put him there? We have to take him to some normal school. I don't think he can do that. But she was the whole hearted, and she said, no, we have to push him. Just give him a try. Let us see how it goes. So I bow my head in front of her, because just because of her trust on me, or a pressurization and push on me, now I'm sitting like this. Otherwise, if it was the other way around, being put up, I might be happening maybe better, or I don't know where I am. So I don't say I might go bad, because parents takes right decision in most of the time, and they know much better. Because I'm talking about an era where there are no these many multiple cable DTH channels. Just we have Doordarshan, and no YouTube, no internet other than land phones. So I'm talking about that era where we are not much known about what is happening outside our town or maybe village, apart from what is being at the 7:30 news in Doordarshan and the next day morning newspaper. And not many newspapers like today, maybe one or two. That's it. So there's all things inspired when we look back. So that's why wherever I go, I used to tell, my dad was one of my biggest enemy until my degree, bachelor's. But after that, he is one of my superhero. The only because he was being in that way, that's where framed me to be to be not so naughty. maybe, right? I don't know. So I used to tell this everywhere. It might be painful to you when they say no, don't do this, don't go, but later you realize when you have the same situation or when you have a kid, you realize this, right? So keeping all this in mind, let me move forward, not too much into personal aspects. So my academic journey, of course, dreamt about UPSC when I was doing my 10th standard, but then prepared for it, then later understood it is not my cup of tea. Okay, then left it. And then when I went with the plus two, somehow that got inspired or by looking at someone or being I'm knowing some a little bit of basic, that time it was basic programming, so was trying to do. And then he bought a PC for me. I started teaching kids and all for games and other things, renting out few things and all. And then I was thinking, okay, I should be a computer engineer. I should program some. I want to make softwares which I can see. That time I was using Windows 95. Okay, not Windows 98, not Windows 2000. It's Windows 95. You have to boot your computer using a floppy disk, the big floppy disk. Without that, it won't boot. And the main thing you had was Minesweeper game, Paint, and WordPad. Nothing like other sophisticated softwares what you see now. So that inspired me to go get into computer engineering. And then somehow I ended up with BSc biochemistry. First thing was I tried for engineering. I told you, know, I was not that great in my schooling. So I did not get a merit seat like NEET like entrance exams during that time. So, and I was not so interested or keen to go with a management seat or something like that. So I ended up, okay, I got in merit because I was very good in chemistry, physics, and biology during my 10th as well as in HSC. But other marks were being got much affected, like history, geography, something that I hate. Sorry, I don't hate, maybe I don't understand, or I don't read. I didn't spend time reading. Now I'm asking my daughter to read, which I didn't do during that time. So that made me to get through biochemistry, and it was really interesting for me because I learned about metabolism, proteins, and many more. So while I was doing the project, I came across a software called RASMOL, and that's where I started visualizing amino acids, proteins, PDB. That's the first thing I still remember, how I got attracted to bioinformatics or drug discovery or molecular modeling. And then I was so happy that I'm trying to shift my gears from biochemistry to bioinformatics because it's something related to computers. So I'm so happy. But then later I realized, okay, it's not the computers or what I was expecting. It is more of scripting, Perl, Python. And during that time, yeah, our internet speed during that time was 64 kbps, not 64 mbps what you have now. If you wanted to write an email, you have to wait at least for 2 to 3 minutes, and there won't be these much ads now popping up, and there won't be too much of graphics content. It's very simple and very simple to send things and all. So, and of course, we had a good access to internet. Professors like Sajita, who is currently at VIT. She was one of my tutor lecturer. Then Dr. Stephen from Baridasan University. Then Jayalakshmi. Then Ajita Shanmugam. Then Raja. Then Shegar. All these people were so supportive when we were getting into this, because something new bioinformatics that was that time. So later I realized that, okay, I'm not a really computational bio guy, because I do not know actual programming that time. Even though we learned during the things and all, and fortunately or unfortunately, I was having the university first rank holder from Bharatdasan University during that year throughout all the colleges under that university. So, but later I decided I shouldn't go there because I was earlier good at chemistry and understanding proteins. Why should I go for scripting? Let me shift my gears again to molecular modeling and bit of drug discovery. But then later I joined as a lecturer in Vinayaga missions for their BTech bioinformatics. I tell that story again, funny things are there. And then for a requirement to be a lecturer, I took MPhil. right? It is a survival to the fittest. That's what we're looking at. And then, as I already decided, after MSc, I then MPhil, and then I did my PhD, which is more focused on computational chemistry, especially focused on medicinal applications. So, that was more decided and focused. But let me tell you, between my MPhil and doing PhD, there was a gap of almost 6 years. I did not do my PhD continuously after my MSc. I started working just because of financial commitments and other things. So I also understand all of you also having the same story. But later comes, I want to tell about the research journey. And when you're talking about academic journey, I'm still learning. Only thing is my platforms have changed to Udemy, Coursera, or edX, or even other platforms. Many of them are offering during my free time on my smartphone, taking them and going on. So learning process is daily going on. Of course, I'm now learning what my daughter is learning in third standard, because sometimes we have to look at the history to refer and tell her the story and to do her project. So I'm going back to my school days also. So learning is continuing throughout our days always. And then coming to research, the first position which I took was as a project fellow for an IGNO project at the University of Kerala with Professor Pavender, as well as with Professor Achut Shankar. So I was there almost a year, and from there, that's where I flew into US. So that was a project to design certain course materials and to also take care of lab practicals and look at few minor projects. And then I was being paid and as an adjunct scholar at University of Florida under Professor um, Alan Katruski. Unfortunately, he's no more. He was almost 86 when I joined the group, and he passed away in 2014 Feb. So, and also to carry out research, learn how to write papers very much, and then cultural shock, but really explored many more opportunities when I was in US. How to actually manage our colleagues, okay? Treat colleagues, how to respect them, especially communication skill. I'm not mentioning communication skill to be how you speak English. I'm very bad at grammar, let me tell you that. But the communication skills helped me a lot to learn from there, because you have to listen to a person. Listening capability is something very important. Give chance for others to speak, listen properly, and then respond. You have to know where to pause. Don't talk too much. So those are all things we learn from there. And also how to write papers. I was having a bad habit of putting a lot of articles in the paper. The too much and the lengthy sentences. So these were already known problems. That's what when the professor was telling the same thing. And the day one when I visited and knocked and went into his room, he was saying, I was calling him, said, Giri, I know you're coming from India. I visited India several times. Uh, Please don't call me sir hereafter. You're free to call me Alan, or you can call me Prof, as you like. You call bro for brother. So you can call me Prof. So I was so shocked because I was not used to that, right? So then it was slowly went on Alan, Prof. That's how it was going. And colleagues, even super senior colleagues also, just the name, but they're very comfortable. But I was never used to that. Later on, we understood. And then I understood the comfort in calling in that way or addressing in that way improved our productivity and teamwork. There was too much of good teamwork. There is no hierarchical problems. There is no thinking of what they might think or you might think. And I have seen, I learned how to say no there. So when professor was asking to do something, one of my seniors said, no, I don't have time to do that. He was fine with that. Here, what happens is, even I have that bad habit earlier. Now, I think now being back to India for almost 6 years now, I also started. We say yes to everything because we don't want to hurt the other guy. And we take it and we delay it. And we don't keep up the deadlines. And we'll say 100 reasons why we got delayed. Even I do the same mistake. That reduces your credibility and that reduces a trust on you. It's a very important thing. All the students should understand, whoever is planning to going to work in large group, whether it is in India or abroad. I duly respect Indian research ecosystem. Because I recently, during my TUT, I said, after coming back to India, I realized I should have done my PhD in India, not in abroad, uh, because of many reasons. Now, then later, because of health issues with the prof, he suggested me go along with my collaborator. So otherwise your PhD shouldn't get affected. So I went to University of Tartu in Estonia to carry out my research and design compounds with significance mostly on QSAR. That's where I started working on. And later, I also being placed as a Marie Curie Fellow at Molcode. We were working on Parkinson's disease to identify lead molecule along with University of Helsinki. And then coming back, became a research advisor to VIT and started guiding a PhD student. I'm happy that next year she is going to graduate. Already published around 3 to 4 papers. Sachida is also one of, her main supervisor in Sci'versation. And now taking the role with Shastra and Nairo. Shastra is more focused on softwares like sales and support and customizations and all. Coming to Shastra was after my PhD, I was hunting for jobs, even academia in India. I wanted to come back to India. Otherwise, I would have taken green card and sitting back in US. But I came back to India because of many, many reasons. Even my wife also okay with that. She was also working as a system analyst at California with Blue Cross Florida. And then she shifted back to Florida also. So we were happy, comfortably earning good money, but then we thought we wanted to come back to India. So when I was hunting jobs in India, I had a lot of hiccups getting through government policies or some other premier institutes. Even when I'm coming down to my state where I hail from, they have different rules how the recruitment process goes on. So I couldn't get through. Some of the companies game in the package was a bit lower than what was a standard, and postdoc opportunities and many were there. But later I decided during an offer, like a job opening, they are starting a company in India. They want a person who to take over the responsibility. When I was in Estonia, I had a good opportunity to go through many of the entrepreneurial skills. That's where I'm going to tell you a bit more here, which could be a combination of scientific and entrepreneurship. And also, excuse me, Prash, please tell me when you have cut-off time. So don't
Correct.
worry to
Please,
stop
please,
me.
please go ahead, Giri. No problem. Please. Yeah. Thank
Please
you.
feel free to stop me.
We're all,
Yeah,
we're all getting enlightened. Thank you so much. Please. Yeah.
thank you. So going through the professional journey, this is something that I have to really tell you why I'm into this business or sales. But let me be very honest. I don't talk business when I meet scientists. I talk only science. My belief is that when you can prove yourself what is the science and what is the knowledge that you wanted to transfer or what you can offer, automatically business happens. You sell yourself or you sell what technology you want to sell rather than talking about business. So my professional journey started with the designing, branding, and sales when I was in schooling. My dad had some agency for advertisements, and he had a local government body tie-up to for like advertisement boards and other things. So I was already started interacting with the customer, but a different set of customer or different category, and then meeting their designing needs, branding, and advertisements. I'm very good at Corel Draw, Photoshop, all those things during that time. That was during 1999 and 2004. Then I left my place. So that's where I started with my professional journey, where I learned from people, learned from my dad also, like, and going again to customers for waiting for payments and all those things happened during that time. Later, joined as a lecturer just after the day of my result. I still remember 1st of June or 2nd of June, I was joining as a lecturer. But there is a funny story there. After all my interview, they made me to take class. Everything went well. And the first day of my where students are coming in, so they had the first batch of bioinformatics that year. Lovely students. They're still in touch with me. They still talk to me. We have a group, and a lot of interactions happen. The funny thing was, I was short in height. I'm still proud of that. When I was entering, walking into the gate, he looks a bit tall and very, very bulky guy. As a first year student, he started ragging me, not realizing I'm a lecturer. So when I said that I'm a lecturer, no, we don't believe that you're a lecturer. We think that you're a first year student. Okay, I don't know him. He don't know me. Then I had the first class that day as an orientation class, being a coordinator for the bioinformatics. When I was going there, he was sitting in the last bench. So unfortunately, we are in the same class. He was so nervous, so embarrassed. I took it as a very funny thing. I felt bad, but okay, fine. We have to go through. This is all new experiences. And during the interval time, he came and talked to me. Sir, please don't take it seriously. It was just, I know it was fun, but it was up to the limit, so it was okay, no problem. So I'm so happy with, they were the first bunch of my students, so happy and still connected, and all are well placed in many, many places in different field of professional life. Later, I joined as a support specialist in Acceleris in Bangalore. And after the interviewer told me, "You're so qualified, too much technical, so we do not want you because you're overqualified." I said, "No. The reason behind that is the salary due for a lecturer during that time without a PhD was too low, and there'll be delays and all those things happen." I said, "I have commitment, so I'm okay to take it. I don't look at of my qualification. I know how I can build up my career." Let me tell you, what I'm doing right now is what I did earlier as a support specialist. So I really being very thankful who reconsidered me. So I came out of the office. I was about to go to the bus stop. I was very close to the bus stop. I got a call again. Giri, we discussed again. Can you please come back? We have to discuss. I went back. They said, since you were so confident that you can do it, even though you're overqualified, don't think I have too many qualifications, just MSc, okay? They don't want MSc. They want only bachelor's or a diploma. That's what their expectation was. But I think they discussed internally, and then they offered me the job right away. So even though it was a support specialist, much more of IT, Linux, Unix, Solaris. That time it was Solaris, Windows support. But later, when they saw something, they additionally gave me about a bit of training of GCG package. I don't know how many of you are aware of it. GCG package is something for sequence analysis. And then slowly getting into Discovery Studio 2.0, that time it was, or 1.7. So not for many key customers, at least for students. So I have to learn and then teach them and support a bit more. But there were many more super senior experts who were really giving the talk, and I don't talk about that, but more of to the students. I was a support specialist. Later shifted to Mumbai. That was a tremendous experience. I would recommend everybody who is not living or metro cities, but especially Mumbai, I should pick it up. You have to travel in that train. Because if you miss a train, even the next train comes within 2 minutes or 3 minutes, still you will be late for your work. So that is the thing. So you have to keep up your timing, and you have to be, you need to know the tactics how to get in and get out of your train on timely and without getting hurt. So time management, how to manage. So you do not want to be so expensive on eating eateries. So you will be preferring street food. For your Mumbai, for my morning breakfast, most of the time I would go for vada pav. Then there was chai, the Mumbai chai, special chai, and at afternoon there'll be chas. So we have to experience that. Don't be so picky that since the food is not good, I'm not going to that place. Since that is not good, I'm not. No, you shouldn't be so picky. You have to go for opportunity. Then only it can come through. And then went to US. There were a lot of cultural shock. Even I was late by 2 or 3 minutes for a meeting by prof with Henkel. And I was late because the buses there, if it is full, they don't allow other people to get in. So I have to wait for the next bus to get in. And it was my only 40th day after joining. So he wrote a big mail to the other team saying that why I was late, all those things. And he told me, Giri, this is US. In India, maybe, I don't know, maybe for you, 1 o'clock, maybe 1:05. But here, 1 o'clock means 12:05. sorry, 12:55. So please be sure that. So he was so much dedicated and focused, so that nobody should get affected, and he was also really safeguarding us so that nobody feels bad about what the mistake that we have done. So that is really I learned from them. So he, rather than a boss, I would call him as a leader. He was in front of us to safeguard for everything. There's a big difference between boss and leader. I have seen a lot of pictures and memes about that, but when we realize that, we get to know about it. And also I had cultural shock. As I said, I had a super senior colleague who was telling, why you need to come here and struggle all this? Why can't you be in your country? Why can't you do things there? Why you're coming here and not doing things productively, giving us extra work, all those things? All things happened. But then I was very happy that I was having the freedom to share these things with Prof. And when I told him, he said, don't worry, Giri, this all, everybody faces it. Now you understood what is something you're missing. Try to come up with that. Try to mingle with people. Try to socialize so that you will overcome all these challenges what you're facing now. And finally, he told me one thing. Giri, don't come to my group expecting standard money or stipend. You come to my or join my group if you want to improve your CV. That's it. Nothing more. And before interviews, he took around 7 referees from me. And surprisingly, to all 7 referees, he wrote an email. Whoever did not reply, he called them over the phone. And the main question, 2 questions were only related to science. And other 8 questions were related to, am I a team goer? How is my learning capability? How is my communication skills? Those were the things. So they see how trustworthy you are to give responsibilities and other how accountable you are. And that's where I came out from there with around 33 papers, which is having a combination of synthesis, bioassay, as well as computational. So among 48 members, we were around 6 of them in computational group. So Professor Katrisky is very well known for heterocyclic chemistry, and a very huge group. He is something similar to Sigma Aldrich. He makes compounds. He has his own archive, so, and sells compounds to other also. So this is something really encouraging to me. Professor who is serving many 48 people, including postdocs and students, and his money is completely coming from corporate companies, not from any government agencies. That's, and whatever money he earned, he never spent for his personal thing. He reinvested back to research. So when he fell ill, he wrote a will saying that, please pay, I'm not disclosing the amount, to all my group members. They shouldn't get affected if I am losing my life. And that's what he was taking care of us like that, anything. So none of us being much affected. Of course, it was big loss for us, but he was still taking care of us. So, and he also donated a good amount of money to University of Florida for specific maintaining his chair for research and conferences. And then coming back, startup, went on with a startup, lot of reading, lot of understanding, lot of mistakes, challenging market influence, how to penetrate into the market when already there are large competitors already within the market, like India, very dynamic market. And usually works almost like 14 to 16 hours initially. And now being at lockdown and being at home, works more time, because if it was an office time, it was very flexible. Now I agree, I think everyone agree with me, you are working more time now. And then more high risk, because if business doesn't happen, my family will be on striving for food. So, and my employee who trusted on me, I have to lay off them. So it's a high risk. It's not only you have to look at your work or look at only your salary, but you have to look at who are all with you, supporting you to move forward. So right, initially 1 year I ran the company just one man show, just myself, no one else, and was able to do good numbers that time. And slowly I started increasing people. Now we are at 48. Why I said 8 to 9, one of the member went for maternity leave. So that's what is happening now. So we need to look at a slow growth, but don't expect tomorrow I have to get the money. That's where we have to look at. And Prash, I have to correct you slowly, and sorry for that. I'm not a chairperson for Drug Discovery Hackathon. I am one of the coordinator for Drug Discovery Hackathon, and I'm sure that many of you will be taking part in it. And I'm happy if there is anything more to be done. I really thank Dr. Anshu Bharatwaj, as well as Dr. Abhay, as well as Dr. Pavandar, who introduced me to the team, and I mean nominated me to the team, and also they made trust on me to giving a great responsibility. Later I started focusing on my always my term, what I look put forward is science for society. Earlier days when I was in MSc, I used to write saying that do science for social relevance, not just for publication. But I removed that when I started publishing. I shouldn't have any conflict there, right? So science for society, if you look my Twitter account, that's where I have written. I know many of them have registered this name for organization, but you're free to use these terms. Okay. So I have many slides in my SlideShare, few technical and demos in my YouTube channel, workflows and models of QSAR in GitHub, and still I'm supporting internships and project guidances to many students, both paid and unpaid. Paid is only because to make them accountable, and whatever they're paying, they're going to get it back if they do a presentation of the project that they're going to do with me somewhere other than their parent institution. They get it completely refunded. And there are more than 68 students have completed internship with me, and many of them have taken the refund also. So I'm so happy because they respected that accountability, because something always going for free, we always give it a chance for tomorrow. Okay, I'll do it tomorrow, it's free. No, there's no deadline, it's fine. But when you have something, you have to be accountable, and when there is a deadline, you have to finish it off. You have no other choice. By the way, so this is my family, my in-laws, my parents, and my daughter. Now I have a son. He's also a little one. And then my school friends, and I don't know how many of you know can see this picture clearly. His name is Professor John Desnoffer. I don't know how many of you know him. He is a Nobel Prize, a Nobel laureate, 1988 for chemistry. He got Nobel Prize for being the first crystal structure of an integral membrane protein. So had a chance to meet and interact with him and his wife when he visited University of Kerala when I was there. And this was a team at Estonia. I hope, Prash, you know Nyaneshwar. I saw recently you have published a paper, so we were all there at
Right.
Estonia, and his wife Chandana. They're all back to India, so I'm so happy. And which I usually tell students also: please go abroad, get experience, get take the good message from there to India, and try to spread out here. Let other people who cannot go to or did not get chance to there, try to help them with the same culture or same other things that you expect. I'm not going to move words to avoid any conflicts here. Even Sandeep, he's also now working in Origin in Bangalore, so most of them are back in India. Others are all, they're still continuing their studies there and postdocs and all. Of course, there are cultural events. We try to introduce Indian festivals, everything in Estonia, and this is the group in US, whom I was telling you about. More than 48 group members with. Of course, family members are also there. So very social and very engaging. And these are my students with. During their farewell, I went and met them. So they're all now in different places. And this is where I was with Pavan sir at University of Kerala. And here you can see Professor Achut Shankar also. They were too really supportive and used to properly give right guidance decisions. And then I was getting into entrepreneurship. Would really thank the University of Tartu, Microsoft, and also the local governing body of Tartu, because whoever you see here next to me is the Prime Minister for Estonia. In Estonia, Prime Minister has not so much of importance, but more of president. So he came up to give us prizes. So we won a prize, and the two projects was Peer Cloud, and the other one was called Student Response System. So we won some cash as an investment to the company. Of course, when everybody went in different places, of course it's still stagnant, but we have not seen anyone has done that so far. And there were a lot of learnings happened: how to kill an idea, how to do value proposition, what is a business idea, how you can pitch it to an investor. So this presentation is I'm pitching to three investors. Of course, we were able to get almost 8,500 euros that time for that project. So these were really that's where, when I went to Estonia, that's where it was really kickstarting with entrepreneurship in my mind. So that's where I combined science and entrepreneurship, and also a transfer of social, cultural transfer, because that also happened. We wanted to introduce Indian culture to the Estonian citizens, and we were so lucky and happy to do that. So he is Professor Alan Katruski and his wife, and they started a charitable trust, and they're still running. These were the few members of a group, and this was my second group. There is one more group where we were working with. So you can see how old he was by then. And earlier also, too many presentations, a lot of delivering of classes. I love taking presentations. I love teaching. I love speaking. But I always tell them, if it is too much, please let me know. I'll stop. So it is very important time. If you go on keeping, even if it is good for longer, it will be so boring. And this was happened recently last year in India. So Marie Curie, they were doing a Eurases roadshow to give an to talk about our journey and experience being a Marie Curie fellow to the prospective students. So they invited me, and I gave my experience as being a Marie Curie fellow at Estonia, and also met many people. So he is Dr. John, who was a Nobel laureate, and his wife, and he is Dr. Andres Lijas from Lund University, Professor Pavender, Professor Valdemar. I don't know how many of you know Dr. John Irwin. He's a person behind the Zing database and also DocSec software. So we met him in India, both of them. So he is Professor Matti Karlsson, one of my PhD guide, also Kaido Tam, and Stephen during my master's in bioinformatics, including Jayalakshmi, Sajita, many more, many people went through my life. And also my hobby and my leisure times, because too much of work is also going to tire you, right? So when I was in US, I started involving with Asha for Education. It was really wonderful experience. So we used to take a stall in for UF Gators. So Gators is a football team, not the football what we do here. It is I don't know the name what we call here. Sorry. So that's the football there. And we used to put stalls. Whatever the income that we get, we put it into Asha for Education. So we used to generate almost 48,000 US dollars per annum and send back to India. Asha for Education is for empowering women education, skill development, and also many projects in Kolkata, Kerala, and West Bengal, Assam, many places we used to work for them. And during my leisure time, all these hiking, kayaking, wall climbing, even dance. Also, we did a skit in US, and later my nickname was Gator Baba. And still I use Gator Baba as my Wi-Fi router's name. So it was so much things learned and experiences, and I would love to travel. So explored many places like St. Petersburg, even in US also made a road trip with my wife. It was really, and coming back to India, I don't too much drive, but I have my bike, and I used to do that. So during lockdown period, I took my bike, the Bullet, same one, from Bangalore to my hometown, which is around 770 kilometers away. So the whole day I was driving. So I like going for rides. That's all from me for today. And if you want to get connected with me, these are my numbers. I'm available in WhatsApp as well as other social handle. But over to you, Prash, if you have any questions. And then one, two questions people send me, I will also address them after addressing your questions.
Yeah, yeah, wonderful, wonderful, Giri. I mean, thank you so much, for this, limelighting journey that you have gone through. I mean, I think this is quite amazing. Thank you so much, for your wonderful presentation. We're all certainly, getting inspired. One of the most important thing, one of the most important things that probably the student fraternity, can probably, take a leaf from this, including me, is that one certainly needs to be a good listener. That is the most important trait,
Yes.
and saying no, of course. And if there is something like negativity, that is always driving around you, always think that you yourself uh, are a positron. That's reason why you are a positive force, and there might be a lot of negative forces coming through you. But always try to convert those negativities into positives. That is a wonderful thing, and from saying no, that is what you learned. And in fact, I also learned a lot on how to say no after having been in Europe for about 12 years. That's quite fascinating. I have about 6 to 7 questions for you.
Oh my God.
Yeah. First, starting with your wonderful education. So, if you really take it forward, Giri, as a computational chemist, there's hardly any molecule, any lead molecule that has really come up successfully in India. Although there are a good number of molecules now that has gone for probably post third phase or fourth phase towards drug discovery. We're not really making a merry when we really look back and ask ourselves that there are many computational chemists. So if you really see all the bioinformaticists today, they have really carved out a niche from computational chemistry stream only. Many of them are really chemists. If you really talk about bioinformatics in India, the first thing that really comes into picture is the drug designing, not really the native bioinformatics or the genomics. So
Mhm.
why is that? Where are we really lagging? As far as Indian bioinformatics is concerned, especially towards discovery of lead molecules.
Yeah. Very great and relevant question. The simplest answer is day before yesterday's Nobel Prize for CRISPR-Cas gene editing. And that prize is for chemistry, but where did they apply it for? It's on genome editing. As simple as it is. So, what is missing here is, uh, no offense, please excuse me for saying this. Our bioinformatics curriculum earlier was more focused towards biology, and later it was too late to realize that we need more of programming capability, especially scripting capability there. And when they realized it, there was not much of openings. Now we have NGS coming up. Coming up means already there. Now also we need data scientists. Today I read around more than 93,000 data scientist jobs are available in India, but vacant. There's no right candidate to take it over. So we are a bit late to realize what is required for tomorrow. Times of India last year, they had a survey on engineers employability, and they found 83% of the students, current students of 2019, are ineligible to get any future jobs. It's 83%, not 2 or 3%. So we cannot blame the curriculum. We have certain limitations, time limit, everything to be focused. So it's we who have to improve. I have seen many students in LinkedIn, they're posting when they get Coursera certifications and all. Good, do that. If you don't need certifications, fine. Do an audit course, free. Do it. Tomorrow, when you go to interview, they don't look at your certificates. Let me tell you, I don't look at certificates. I look whether you have a degree or not. That's it. I don't look at your marks. I look what you're going to know and what you're going to address when I ask you questions. If I'm asking you to write a code for this particular problem, if you write, you're done. You're good. I don't look at what you got marks. So it's not about marks. It's about what you can do, how worth you are for the company. And coming to the other part, molecular modeling. We think that drug discovery can be done by a bioinformatician or a computational biologist. Of course, we learned about it. But please I'll be very frank, you cannot do it. For drug discovery, you need understanding of organic chemistry. Means just doing an analysis, just doing a docking. Of course, you don't require any chemistry understanding. But that is not a drug discovery. Discovery means what? You want to optimize it. You want to make modifications. If you simply change methyl group to hydroxyl group, of course, your activity might increase in computers. Now, the question is, can you make it in the lab? No, you don't know because you're not a synthetic chemist. You're not an organic chemist. So you have to either buy or talk to a chemist. Now, he tells you, no, this cannot be synthesized. But you tell me, no, this gave a very good activity computationally. Okay, but this cannot be synthesized. So there's a big gap between two different fields. It could be computational modelers, chemist, biologist, bioinformatician, whoever, with chemist or medicinal chemist or organic chemist. When these two guys work together, then only there is a complete sense of drug discovery. In earlier days, when during my graduation and all, there were only few companies in like Osimum Biosolutions, Sci Biosciences in Chennai, GVK Bioscience, GVK Agriculture, I think that time during at Hyderabad. There were only hand-picked companies here and there, and they also did not later did not take any bioinformatics because the curriculum was not meeting their requirements. So if anybody wants to get into drug discovery, change your track, get into chemistry. I don't say pure chemistry, but you need to know. Now, when you say that, do I need only chemistry? Sorry, no, you need to understand the mechanism also. Because that is why we have a new phrases being around recently, like around 4 or 5 years, mechanistic driven drug discovery. A drug is not of use, right? You need to know how your drug is going to react with your organs and how your organs are going to react with your drug, vice versa. So you need to know the mechanism. You need to know the metabolism. So until unless you know the mechanism, how can you design a drug to match the mechanism or to being agonist or antagonist, or whether to inhibit or whether to activate? So you need to have a understanding there. It's not about degree. That's where I didn't get a job in academic in the government sector, especially in my state. They said, masters, you have learned bioinformatics, but your PhD is on relatively computational chemistry. We have an opening for computational chemistry. Since your master's in bioinformatics, we don't take you because we look at the major topic, what is it you have taken for masters. Different state to state, things are changing. So first thing, we have to have change or updation in policies because they are making new courses. So change the policies accordingly. Everybody is capable. Give them an opportunity to learn. That is very, very trivial. No, don't say no to everybody. Of course, if it is too bad, otherwise you know them that they can do it. Give them a try. Give them a chance to learn. And third, don't think that I cannot do this. This is not my area. Let me leave it off. Okay, if you're bioinformatician, for example, sorry if I'm pinpointing Gnaneshwarji, who is now in Banaras in the university in zoology department. He has very good, wonderful publications, and mostly in genomes, evolutionary studies and all. Lot of BLAST and Perl scripting has been done. He is a core user of it and also does a lot of customizations. He is a bioinformatician for me. Second, our Prash, he also is into the same domain. Now, if you ask me, I'm not. I might have learned bioinformatics, but I switched my area because I'm more focusing on molecular modeling. So we have to have an understanding. These two are related but different. You need to have different perspectives on understanding both of them. I hope I gave a good understanding about or an opinion about your question.
Wonderful, Giri. I completely agree to that. Yeah. Second thing, Giri, I think, as a chemist, you have worked on various aspects of azoles. And I mean, right from your heydays and as well, during your PhDs. What aspects of, QSARs have really, created a kind of an interest in working on azoles? Is it because of your wonderful collaborations with your PhD mentors, or is it your own ideation that you worked on these number of azoles? Yeah.
Yeah. So hello. Yeah.
Yeah, yeah,
So,
please. Yeah.
yeah, aerosols, we worked initially as a collaborative project. When I went there, we were about to get like experimental data from US Department of Agriculture, whom we were working with, like mosquito repellency and all. So aerosols were that time from collaboration from chemical engineering. So we were exploring about the air pollution. Even aerosols also have other properties. So computationally, what we tried was what are the stable conformations which could be achieved, and what are the physiochemical properties that we could predict on the aerosols, like being it is a solid particle or a liquid droplets. We wanted to see how far it is going to be affecting with the pollution. So that way, the thermodynamic or statistical thermodynamic properties could be predicted using QSAR. So that was the first paper which I worked on aerosols.
Yeah, wonderful, Giri. Yeah. So, Giri, if we see the SAR models, I mean, whatever SAR models that have been vividly used for development of machine learning approaches, there is also something called SAR paradox, which means we always think that not all the molecules would have probably the similar activities. So,
Yes.
so given the current challenge, especially given the current COVID-19 drug challenge, how do you see or how do you take up this particular SR paradox during the next few years?
You rightly said, all similar molecules will not have similar activity. That's what I also pitch in wherever I present, because most of the time we usually do a similarity search or cluster analysis using an FDR as a reference compound, and then pick up the molecules and then start our research on some way or the other, X and Y, extra, etc., etc. But what we should understand from that is same similar compounds will have same physiochemical properties or molecular properties, but cannot have same biological activity or potency or same or similar pharmacokinetic properties. A change of a single element or a single atom is going to tell you whether this is metabolically stable or not. Just a change in a single element. So for that particular compound, your similarity will be anyway around 95%. But change of a single atom is going to say this compound is for drug strategy. It will get broken down during liver metabolism or some other metabolic pathway. So even coming to COVID also, that is where the challenge. We are nowhere. We are talking about many stories. We have many positive expectations, but still we have not reached anywhere. Of course, vaccine we are expecting somewhere in the next year, early next year. But otherwise, in the drug side, we are still in bias. So we are going for a combinations of different things. So when you're looking at SARs, you have to be very careful. That's where what I'm trying to tell you. You need to have an understanding about pharmacokinetics, about and also to understand the structure. Earlier days, people, especially scientists, they don't require any computers. They just look at the structure and they say, "Hey, this is having problem. We have to replace this." Now, why we are using softwares? Because we are not expert in that. We are not understanding the concept, or we are not having thorough understanding about different functional groups. That is what my take on that.
Wonderful. Yeah, wonderful, wonderful, Giri. Yeah. So certainly moving towards your entrepreneurial journey, Giri. I know you have amazingly pointed out, what made you to take up an entrepreneurial journey and all.
Mhm.
But not all scientists take up the failures and convert those particular failures towards success stories.
Mhm.
And amazingly, I believe from your vast experience that you have converted these wonderful failures to opportunities and successes. So,
Mhm.
what best advice would you like to give to the young brethren over here on how to take up these challenges and convert those particular challenges to success stories?
Yep. Poor broadcast. Thank you so much for that. What I wanted to say here is when what I was thinking was getting into academic, do research, and I want to transfer what I learned from other places, the good messages. If something is lacking here, try to help them and come up with that. But later I thought, if I go into an entrepreneurship where I can serve many institutions like that, so why should I feel bad about it? For example, if I got a job in an institute called X, I will be able to teach only the students of X. I have restrictions to take classes for others on a regular basis, or I cannot take a project for. Of course, I can take intern students, or I can be invited speaker for conferences. That is fine, but otherwise, I'll be always restricted within that particular institution X. But what now I am doing is nobody is there to ask me, don't do that, don't do this. What is really ethical? I'm trying to do it. Now I can teach many students, many institutions. Many institutions can invite. No ego problems. There, here, go there. If you go there, you cannot go there. Nothing like that. So entrepreneurship gives you the freedom especially on the scientific side. Gives you the freedom if you have a passion of teaching and transfer of knowledge. Nobody is going to stop you, and the others automatically invites you. You need not to write an email. I am free to give a talk. Do you have an opportunity there? I can give a talk. No, you need not to write. Automatically, people will invite you. So that is one freedom of entrepreneurship. Second freedom, you try to meet a different types, levels of society, and you have to meet them. In my company, whoever comes in, even students comes in, I can see some of the students are already attending this session. I have told the first thing is once they decided that they're joining us, don't call anybody here within this company sir or madam. Just ask their first name or last name, whichever comfortable, call with their name. It will be difficult, but later you'll be more flexible. The only reason was, again, I'm talking about entrepreneurship. The students are going to tell you feedback. Sir, the way you presented was really good, sir, but I didn't like that. That is really important for me to improve or others to improve. Appreciations are always good, but you need to know what is bad. Then only you can improve. That's where we have to understand. You never should confuse between what I like and what I understand or what is easy. What you understand is what is always easy for you, not about what you like.
Yeah,
Be
wonderfully
very
insighted,
sure in that.
Giri.
Yeah.
Yeah, yeah. Please go ahead. Please go ahead, Giri. Yeah.
Yeah. So this was taken from a book written by Malik. It's a German book, but I got a translation. So that's what we have to understand. For example, plus two, if mathematics was too difficult for me, then I will say mathematics is hard because I didn't try to understand it. That's what we have to realize. So you have to try to understand. And the next part is that in the company, even my students used to give me a feedback. I used to take an opinion from them. And very important fact is try to acknowledge whatever they did for you. So when I go for presentation, if they did some of the, I say, this is my student who have done it. I didn't do it. Adding a 2 second extra phrase is not going to harm you. My student will be so happy that I have acknowledged them. So even though I'm talking about entrepreneur, I'm still talking about students because I employ also the same story. There's no difference between a office boy in my office and whoever a manager in my office. All are of same category. They might have different degrees. And let me also disclose one secret. In my company, a degree holder is getting lesser price, lesser salary than a non-degree holder. It's because what they're delivering to the company. It's not about degree. So the person will be given the right value and right recognition and right place when you're going for entrepreneurship. And it has its own risk, of course. You have this risk. When I started, I put my all my family savings, what I earned in US and Europe. That's where we put into the company, including some investors were there. Now, no investors. We bought back all the shares. So we have to be independent, and you have to be ready to take the risk with more vision and focus. You can make mistakes, but mistakes should be only once. If you repeat the mistake, it is going to make you a loss. So you should be very carefully going. Take some time to read, talk to many people, take opinions, and then make a decision. Don't get biased. That or this, then tomorrow morning you make a decision. That might be wrong. So take different perspectives and make decisions. Yes, Prash.
Right. Thank you, Giri. Giri, Uma Kanga is an assistant professor at All India Institute of Medical Sciences, Delhi. She's a transplant immunologist. So she has a question for you. So she's here. Yeah, please,
Yeah.
ma'am.
Hi, ma'am.
Good evening, Dr. Giri.
Good
It
evening.
was a pleasure to listen to your life journey. I don't have a question for you. You have done so well in life. Just because you're teaching to the students and giving them good messages, I wanted to add one point that what I, from your, that when you mentioned your initial slide that you appreciate the people who said no. So I just remembered some quotes from our ex-president, Sir Abdul Kalam ji. And I wanted just to share that because there are your students, and I'm sure as you learn from those no, there are a few things that I wanted to say that what
Please.
he mentioned was no means next opportunity. So I
Oh,
think
yeah.
the students who are present here must not take no as a bad. It means that life is giving you a next opportunity and we should always grab it. Like
Absolutely.
you are a living example for that. Other things that I wanted to say was when people say this is the end of life or this is the end of my this attempt that I was doing, there is never an end. End means effort never dies. We should take it as effort never dies. So that end is not an end actually. We can go
Oh,
on make more effort.
wonderful. Wonderful.
And one more thing that despite all this, if we fail, so if you take FAIL, I have followed the motto that this is the first attempt in learning. So
Mm.
FIL, none of the students who are present over here should take it as a failure. It is a first attempt in learning. We can also take it as find another alternative in life.
Ah,
So it's not necessary that what we are doing is the right. We can always find another alternative and we can be successful. So by your, that know that you got from people, I think you made a good outcome in your life. And I wanted the students to know this thing. So I follow these three things, what Abdul Kalam ji, our president, said, and I just wanted to share this. And
thank you
appreciate
so much. It was really great.
all your contributions to this scientific field. Thanks, Prash,
Thank
for
you.
giving me an opportunity
No,
to.
wonderful, wonderful, ma'am. It's so nice. I mean,
Yes,
it's absolutely wonderful. I also got to know this. I also noted down in my notebook here. Thank you so much.
absolutely.
So, so that comes to my questions. I also have another question, probably which I'll ask at the end. But Giri, we received about 43 questions, but most of the questions were towards career development. But one of the
Mhm.
questions that a candidate asked is, unfortunately, whenever bioinformatics is considered as a kind of a tool
Mhm.
or computational biology as a discipline,
Mhm.
only less than 10% of the tools are absolutely perused by researchers,
Mhm.
for an effective sense.
Mhm.
How do we overcome this particular thing so that users can better take up opportunities and making these tools for viable use?
Yes, a very valid point. Here I have to repeat again. Don't confuse between what I like and what is easy. We are always looking forward to what is easy. One, we always look at what others do. Then we have to do the same thing. Third, we always refer to existing knowledge. We never come out of the box. Rather than saying we or generalize, let me say I. Okay, so I don't want to get into any conflict. So we look into all those aspects, right? Whenever students approaches me, even recently also, sir, is there any easy topic I can do? I have to finish the project soon. That's the first question comes in. There are many students who says, sir, I am very passionate, I read about this paper, it's really interesting, so I wanted to work on it. I said it might take a bit more time because a lot of literature data collection, many more analysis is there. So it's okay, sir, I read about it, it's really interesting. That should change. So they liked it, they understood it. Whatever it is difficult or time taking, I have to do it. I know constraints are there. The problem with me, or again, as I said, we or I, me, we try to do everything at the last moment. Tomorrow is a submission. Today we sit and do it in the last. Already 30 days would have passed. It has been given 30 days to do. So that is, we have to prioritize things. Good time management is also very important. Second, we said, I told you that what others used. I'll come to the point of the tools. What others used to be used. So we always ask, hey, which is the right one? They'll say X or Y. Okay, I will also use X and Y. We never analyze whether that X and Y could solve our problems or whatever problem statement you have in bioinformatics or whatever field. You never analyze. You said, oh, he's a right guy who said and recommended I'll use that. And the third, existing knowledge. Let me be very open here. QSR papers, if you're taking 1,000 papers, among 1,000, only 2 to 3% or maybe max 5% is reproducible. Others, all you try to do reproduce, you won't get the same data. It will be something different. So when you publish a paper, it is about reproducibility, reliability, and how much you can take from there. The brain of the paper is always your discussion part. What is your take-home message there? Until unless you read that, what's the use? So until it's reproducible, reliable, there is no point. So as I said, 3 to 5%, let me take in that way, is reproducible. Others are, but we are not finding the right one because we're not testing it. So whenever I request my students, please go through that paper, reproduce the data. If it is good, you take it. Otherwise, don't take it. Especially on predictions, not only QSR, let it be docking also. So now I'll answer the question for the tools. Now you know the context. Why we are not using only 10% is used. First, we are against scripting. We are against Linux. We are against a command line. Wherever there is a GUI, we jump in there. Then that is the best software. I don't know how many of you are using Rosetta here. How many are using Modeler here? These are the two accurate, best protein homology modeling or ab initio modeling tools available globally. Even commercials are below that. I don't know how many of you are using Autodoc Winner. Popular, most of you are using. It has its own drawbacks, but that is also a top accurate one compared to others. On certain cases, not all, okay? I'm not generalizing it. Coming to molecular modeling, dynamics, GROMAX, AMBER, NAMD, these are all there available. But how many are using it? We are still looking for easy way. Where, of course, it is a process by process. When you're coming to bioinformatics also, earlier days, there was something called as JEMBOS package. I don't know how many of you are using it now. JEMBOS. I have used it. So those are step-by-step process. We will have a bundled local blast DB and then do a local search rather than online search. Because earlier days, internet was a difficult, right? We keep it for download and it will download and we have a local repository. We have to analyze any tool, any software. First, you have to evaluate it for your research problem, not for example problem or not for the tutorials or not for others problem. What is your problem? Evaluate it. Are you able to recreate it or are you able to validate it computationally? And then that is the best tool. We are not doing that. So if I say compound software X is being used and you can publish in high grade journals, I don't think so. I have published using Autodoc in 9 impact factor, even 14 impact factor journal. No reviewer asked me why you did not use commercial tools. Nobody asked me. So nothing like that. It's all we are creating it. So for me, tools means it has to meet my needs, my research needs. For that, you have to test it. If you look at my papers, different papers, I've used different tools for QSR. Do you think the other softwares I have not used? I have used, but it did not work there. Their parameterization did not work for that particular data. So I couldn't use. Recently, I published with the PhD student publish two papers, we use two different tools because the same tool did not work for both the problems. So we need to evaluate and understand. Then automatically there will be a normal distribution of usage of tools, and try to reduce the observation saying that I am against command line, I am against scripting. Come on, that is what is data scientist now. Whoever data scientists who are being paid 30 lakhs, 35 lakhs per annum, they are working on Python. So you have to now decide whether you have to say no to those things or yes to that right now, because the time that you're getting now to learn, you will not get when you get graduated. After getting graduated, the parents will start asking you, when are you going for job? Friends will ask you, haven't you ever not got any job? Other people will be asking, what are you doing, dude? That question is going to really make you worry. And that time, if you sit and learn as per the market requirements, you'll be stressed, not so focused. Some people can, others cannot. So it is now it's the right time. You have a lot of things that you can learn. Now I don't tell you because too much of online classes and meeting. When the lockdown started, I am sure you had a lot of time, but now it is too bad. But try to find some time and upskill yourself. Upskilling is very, very important nowadays to get a proper job. That's all, Prash.
Wonderful, Giri. I mean, amazingly inserted. Thank you so much. Giri, just for brevity of time, I'm asking, three more questions, from the registrations. So if
Sure.
you allow me. So one of the important questions is, how does, Giri get self-motivated? His life is, full of illusions, is full of, negativities, and he has really wonderfully taken up, the demotive force and successfully converted that particular unmotivating force to motivation. How does he take up? That is, a question, from one of your students. Yeah.
Uh, Thank you for that. Rather than motivation, I always being down. That is very true. I'm also a human being, so I was being very down after knowing that. But I'll forget about it because getting into other responsibilities. But quickly a day, I realized why that no came. Something because I'm lacking. That's why. But how the motivation happens is when I see my family. One. Second, when I think about my commitments. Third, the girl who came believing and trusting me, and the dad who handed over her hand on my hand. For a married person, right? But for an unmarried person, probably you might think about the dedication, the time, and all the efforts put forward by your parents. So when you think about them, automatically you think, okay, I shouldn't fail. Somehow the time that they have spent on us, we have to give back, being we are successful in our life. It's not about giving back money or looking after them. Of course, that automatically happens. But what I'm trying to say is, if we are successful in life, that is what is a give back for our parents. And if we are successful to the trustworthy, our partner, our a spouse, that is also a give back. So that motivates me when I'm thinking about them. About me, of course, I'll be always down. I am not good for anything. I'm always getting that, oh, you're not good there, you're not good here, but you're good there. So you do that, this, then. So a lot of comparisons coming. But when we think about who are along with us, we also have a responsibility to go along with them and making them to come along with us. That motivates me a lot. Second motivation is when I look at my smiling people. I always try to keep smiling. I very rarely get angry. So that really helps me to keep self motivated.
Wonderful, wonderful, Giri. Next question is, if we really look back in India, many PhD fellows struggle with not so friendly PIs or principal investigators. What is the best advice, for the budding PIs who can be good mentors or guides, for their PhD fellows?
Hope it won't be controversial. Okay, let me try to say that. Uh, I understand this question. I have listened to many others telling the same story. But we have to see our patients. One thing we have wanted to know is, that's what I said, no communication skill. We have to be bit patient, but too much of patience is also bad. The biggest problem with us is basically we think 100 ways before that is happening. For example, if tomorrow I want to ask my dad, dad, can I go for a movie? You will think that he might put some conditional thing, or he might say no, or he might refer to something else, and you will never ask. That is what is happening here. But you are not doing this. Okay, let me ask, whatever it happens, even if it is no also, okay, we are expecting it, let it go. We are not doing that. You have to ask in the right manner, because in India that is very, very important. Right manner, right body language. We were come used to that. There are good things, bad things about it, but let us take the good things. So in the right manner, right timing, ask. Usually people used to say, until the baby cries, the mother will not feed milk to the baby. Then only they realize the baby is hungry, or timely fashion. The same way applies here. Until unless you speak, until unless you communicate in the right way, they really don't know. So it is not about India, it's not about US, it's not about Germany, everywhere. But let me tell you very frankly one thing here. We are in our born country, our home country. The comfort that you are moving around outside your house in your country, the same comfort you will never get in a third national country. So if I'm going to Estonia, or if I'm going to Europe or Australia or US, wherever, there will be a person to ask you, why did you come to my country? Why can't you go back? Of course, it's not going to affect you. You're on your own way. But they are concerned being you're there. So you can do better things here. Why did I come to back to India? I'm still earning good. I'm still running and my life successfully. People who used to ask me, you are being in US, why can't you be there? You could have got green card, bought a house. It's not about that. It's more of freedom. What you can do it in your own country when compared to others. So whatever PI issues all you're facing, try to talk to them. I know only some PIs here, most of the PIs have a social relationship, means taking out, going for a tea together, or inviting you guys to home for a chit chat or a dinner or a potluck. Nowadays it's happening, I could see. Early days, during our days, not much. It was in a different way. I don't want to comment that way. So that I can understand, but now it is a bit open because they're also seeing other people who are doing it. So please try to be open. One. Second, in India, you already know things won't happen quickly. So if you order a chemical or if you order an equipment, it takes its own time, and people never cares about the accuracy. People only look at most which is cheapest. That's what it's just because of our policies. So we have to be prepared, proactive, understand what is going to be done day after tomorrow. Try to do it or prepare for it yesterday, not today. If you are doing in that way, I think majority of the problems you can speak, communicate, and reduce that. Since we are not communicating in the right time, that is very important. Now you might be saying, sir, no, I already discussed with my peer, nothing is happening, because that is too late. And don't compare. That is also happening. Sir, they are doing this, they are doing, why we are not doing this? Don't do that. They don't like, or I don't also might not like, but outside I won't be showing it. The reason is we have a different pace, or maybe slow, or different restrictions. So we have to go along with that. So all these analysis you have to do before joining the PI. So in US you have a in US and Europe you have something. When you get admissions, the professors will be giving presentations of each group what they are doing, and then you can go and meet those professors, one-to-one meetings, and discuss about that. Then the student chooses the professor. That is a standard admission procedure comes in US. In Europe, sometimes it's the other way around. You first contact the professor, get the thing, and then only you go for the admission. So different country, it is different. So here it is anyway, all are public publications, paper, their profile, CV, LinkedIn, everywhere. So try to analyze, understand them, whether your taste, their taste, everything is going matching with the research objectives, and many more things you have to go. It is very true that it's not only about science, it's about social life balance also required to for a successful completion. Thank you. Thank you.
Excellent, excellent, Giri. One final question from one of the participants.
Yeah.
Is it possible? What is your opinion about using machine learning for screening large databases? Are we really ready for it? I believe this is for rational drug designing. That is what I believe
Yeah.
the question is meant for. Yeah, please.
Yeah. We have to first decide whether you are going to be a machine learning software user or a algorithm developer. That first you have to make a decision. If you're a developer, don't think you just knowing Python you can do it. No, it's more about statistics, mathematics, calculus, regression, probability, and many more. Even linear algebra too. Now you might think, oh my God, I shouldn't go to machine learning developer. No, that is a fact, because when you're making algorithms, you need to know mathematics. How can you build an algorithm otherwise? So when you're talking about machine learning developer, that is the case. Now I'm not going to concentrate there much because we are more of bio audience. So when you're going to a machine learning user, yes, it is. Machine learning is really helpful for data pruning, like filtering data. It is going to learn from that training set and going to help you to pick or a decision making. That is really helpful. But I don't give a hype on AI in drug discovery, but I do respect and hype in machine learning in drug discovery, including rationalizations and many more. But don't think that whatever you're doing right now, like in molecular dynamics, all those things can be converted to machine learning. It's a bit challenging because it's more complex. Machine learning is also equivalent to QSAR. Earlier days, during 2014, I have published a review on machine learning in drug discovery. So that time we only talked about QSAR, regression, support vector machine, neural networks, and all. Now it is a hype. Everywhere people talk about machine. Everybody puts they know machine learning also, but it's about how you can create a algorithm. So it is the future. There are many openings available even in bioinformatics and even cheminformatics and drug discovery. Whoever knows about Python libraries as a user, but as a developer, of course, you need to know good statistics as well as mathematics algorithms.
Wonderful. Thank you, Giri. Thank you so much. So before we say goodbye and before I give my
Prash, sorry,
final.
one question was there in the message that they sent. Can I take that? Sorry.
Please, Giri. Yeah.
Yeah. So the question was, there was a lot of hype happening on the diagnostics for when this COVID era started. And there was a lot of misunderstanding and a lot of marketing, everything has happened. Now there is no news about anything about it. So the question was, how do we should expect in the technology improvements after the pandemic situation? That was a question. So as per the recent news and all, I can see many started investing in India. Many started, many industries, manufacturing units started in India. So there is a good go for both for job opportunities as well as for starting a company. The only thing that is, we need innovative minds. We need not to repeat the mistake minds, and also we need we do not want copycats, because that was happened in the diagnostics field. Some of the companies, I don't want to name them, with proof they have already been reported, they have taken the kits from abroad and they modified it and they sold it here. So we have to be very careful. We do not want to do that. We have wanted to innovate. You innovate yourself. So there are good opportunities on the technology improvement that is going to happen. So all of you spend your time very proactively and productively to get ready with the requirements of what is they're going to face you for the tomorrow's employability. That's all. Thank you.
Wonderful, Giri. Do you want to take up any more questions from those questions
No,
that I sent you, Giri? Yeah.
no, that's all. One, just one. That's it.
And do you have any final say, Giri, before I come up with final comments? Yeah.
Yeah, the final say is be patient up to what you can, the limit. After that, please ask. And as Madam rightly said, please, I have also noted down those words, and try to print it, paste it on your wall, and see that whenever you feel down. and be always positive, have always a smiling face, so that others don't feel that you are down. They also make your positive energy. Positive energy and work-life balance is really required you to be successful. That's all. Thank you.
Thank you. Thank you so much, Giri, for your wonderful time and energy today. I mean, you have spent more than 40 minutes today. It's been quite amazing. I always wanted to have this conversation with you. You have been absolutely an amazing person ever since the COVID erupted in India. You started online events. You brought in a lot of assessments about COVID-19. You brought a lot of students together virtually. That has been an incredible things. There were a lot of questions posed. Many of those particular students were really benefited. You're always a student's mentor. I mean, I could always see that having known you for over a decade. And what is most important thing is your down to earth personality. I mean, they never say a die attitude. The kind of personality where everyone would really want to instigate upon and ask that this should be a scientist like personality that I really wanted to take upon. This is quite amazing. So, friends, we all should thank Giri for his wonderful time and energy. He has been an epitome of entrepreneurial success, where he has converted his academic setting towards entrepreneurial journey. He has never said no to the failures. That is the most important thing for a successful scientist. Every scientist would undergo failures at any given point of time. He has always taken the positives and negatives in an equal way. That's where Giri has really taken the shape. And most important thing is, if you really see the kind of computational chemists that are driving Indian research today, there are hardly very few people who can really come up and say that, why is that we are really failing with the lead molecules? And Giri has answer, which he has wonderfully addressed. So that's it for today. Thank you so much, Giri, for having in conversation with me. And most importantly, Giri can be reached at these particular addresses in LinkedIn, YouTube, Twitter, and Instagram, and also at his email, gpillai@zastrine.com. He's a wonderful listener. He's a very good listener. He's a very fast responder as well. And someone asked if there could be any job prospects, associated in his company. You're welcome to contact him. He would probably, have an interview session, with you. And there are also some questions about the job prospects in Germany and other places, which I'm sure, Giri has already addressed it earlier, and we through Bioclues have also addressed it earlier. So in Germany, or for that matter, at any point of time during the COVID times, probably it's not suddenly a good idea to go overseas right now. So, as Giri has rightly pointed out, India is certainly a dream destination today to do good PIs. There is to become good PIs, to work with good PIs as well, because there's a lot of brain drain. Many people have really come down and back to India. They've established good research groups over here. So it's time we developed India. We ensured that we do an effective PhD working over here, and in turn become good PIs. So that's it in Sci'versation with.
Thank you so much, Prash. It was really. Yep. Thank you.
Yeah, thank you, Giri. Thank you so much. So, friends, thank you so much. Yeah, thank you for joining. Yeah.